A key difference in the phospholipid bilayer is the electrical difference when we consider the extracellular fluid side versus the intracellular fluid side, which remember is also the intracellular fluid. So the only thing I want you to know at this point is the difference between the anions versus the cations. So, first of all, it's important for you to know that there is more sodium ions in the extracellular fluid compared to the intracellular fluid. So, for example, on this slide, you can see 8 sodium ions, whereas there's only 1 sodium ion on the intracellular fluid side. Another significant difference is the anions on the extracellular fluid side. Notice that there are two chloride ions, and there's no chloride ions on the inside. Regarding the cations in the intracellular fluid, we can see that there are about 9 potassium ions on the intracellular fluid side, whereas there's only 5 on the extracellular fluid side. Another significant difference is that there are protein anions, which are usually in the form of amino acids, which are on the intracellular fluid side, and none on the extracellular fluid side. Now, the reason that this is so different, so important, is it sets up for a chemical gradient. So there's a concentration gradient, which has already been discussed, plus there's an electrical gradient. And this electrical gradient sets up what's called a resting membrane potential. And so what's going to occur is that potassium is going to diffuse down its steep concentration gradient via leakage channels. So we know that plasma membranes are highly permeable to potassium. This results in a negative charge on the inner surface of the plasma membrane. So when we look here, you can see I'm highlighting the negative charge on the inside of the plasma membrane. Potassium also moves into the cell because they're attracted to the negative charge. Opposites attract. What this ends up doing is creating this negative membrane potential of negative 90 millivolts. So essentially, the way I want you to think of a plasma membrane is analogous to a battery. A battery has two ends. It has a positive end and a negative end. The same is similar to the plasma membrane. The extracellular fluid side is positive compared to the inside, which is negative. And what this means is that the membrane has what we call polarity. And this is going to be important when we get to the nervous system. But at this point, I just want you to know that there's a difference between the inside and the outside. And that difference is that the extracellular fluid has sodium and chloride. Again, when a patient goes to the hospital, they get a saline bag, which is isotonic to their cells. Also, the intracellular fluid, which is the cytoplasm, is going to have potassium as well as negatively charged protein anions. So that's at this point all you need to know about these charges. Our next slide now goes into parts within the cell. So remember, the first thing is cytoplasm, and cytoplasm is made of two major things. It's made up of a liquid and organelles. So in chapter 2, you learned that it is a type of colloid, that when light is shown through cytoplasm, it's going to scatter light. So the solid structures are organelles, metabolic machinery structures, and each has a specialized function. They can either be membranous or non-membranous. There's also inclusions, which are insoluble molecules. They could be things like glycogen granules, pigments, lipid droplets, vacuoles, or crystals. So that makes for the solid part of the cytoplasm. The liquid part is the cytosol, which is the gel-like solution made up of water and soluble molecules like proteins, salts, and sugars. So the membranous structures that are found within the cell are mitochondria, endoplasmic reticulum, Golgi apparatus, peroxisomes, and lysosomes. And non-membranous structures are things like ribosomes, the cytoskeleton, and the centriole. And we're going to have slides on each of these for the rest of the chapter. So membranes allow for compartmentalization, which is crucial to the cell function. So we're going to begin with the membranous organelles. Organelles, remember, are kind of like little organs. And they're little organs because they have a specific function that they play within the cell. Our first slide is going to be on the mitochondria. Mitochondria are referred to as the power plants of the cell because they produce the most important cellular energy molecule called ATP. And they do this with the help of oxygen. So we say it's aerobic cellular respiration. And interestingly, the mitochondria are actually enclosed by a double membrane, an inner membrane, which has many folds and has what are called cristae. And the cristae are embedded with membrane proteins that play a role in cellular respiration. The mitochondria contain their own DNA, RNA, and ribosomes, which makes them very unusual types of organelles because they have DNA. Really, the only other place we find DNA is in the nucleus. So they resemble bacteria. So within the mitochondria, the cristae are located in the inner mitochondrial membrane. There's an area called the matrix, which is where a certain type of metabolism happens. Remember that metabolism is the sum of all chemical reactions in the cell, which is very significant. You'll learn more about metabolism next semester. So there's enzymes that are found here. There's mitochondrial DNA, ribosomes, and an outer mitochondrial membrane. Our next organelle is that of endoplasmic reticulum. And endoplasmic reticulum, there are two different types of. There's one called rough endoplasmic reticulum, and it appears rough because is studded with attached ribosomes. And what's significant about a ribosome is that they are the site for protein synthesis. So a ribosome, if it's free floating, its function is also going to be protein synthesis. Smooth endoplasmic reticulum, on the other hand, is also a network of looped tubules continuous with the rough endoplasmic reticulum, and it has enzymes in its plasma membrane called integral proteins. So when a protein crosses the span of the entire membrane, we call it an integral instead of a peripheral protein. So some of the functions of the smooth endoplasmic reticulum would be things like lipid metabolism, cholesterol and steroid-based hormone synthesis, absorption, synthesis, and transport of fats, as well as detoxification of certain chemicals like drugs and pesticides. So any part of the body where some of these important chemicals are made, those cells would have more smooth endoplasmic reticulum. So if we looked at the histology of a cell and saw more of this, we would know that its physiology would be that of lipid metabolism. Again, the anatomy telling us a little bit about the physiology.